Good afternoon, everyone. I'm Hwijin Seo from Gyeongsang National University. Today, my presentation topic is microbial electrolysis cell anaerobic digestion with intermittent low-strength ultrasound input. Due to the increasing populations and quality of life, treating sewage sludge became a big issue. One of the solutions to treat sewage sludge is anaerobic digestion. Anaerobic digestion consists of hydrolysis, acidogenesis, acetogenesis, and methanogenesis. Anaerobic digestion produces methane, which can convert to heat and electricity, and reduces sludge volume eco-friendly. However, AD has several drawbacks such as pH imbalance due to a different reaction rate between acido or acetogenesis and methanogenesis, taking a long retention time of more than 20 days due to the slow hydrolysis rate and the methane-producing pathways of acetoclastic methanogenesis reaction. And the performance difference depending on the substrate type such as lignocellulosic or food waste, and low methane productivity by methane-producing pathway. So microbial electrolysis cell with anaerobic digestion, MEC-AD, is a combined concept with bioelectrochemical systems and conventional AD. In the anode, exoelectrogens oxidize organic matters, produces electrons, protons, and carbon dioxide, and are transferred to the cathode. At the cathode, the electromethanogenic archaea receive products of exoelectrogens, reduce carbon dioxide, and generate methane through direct or indirect electromethanogenesis reaction. The direct electromethanogenesis reaction, also called direct interspecies electron transfer, uses electrons, directly reduces carbon dioxide, and produces methane. On the other hand, the indirect electromethanogenesis reaction, also called hydrogen-trophic methanogenesis, uses hydrogen or formate as an interspecies electron carrier and produces methane. Theoretically, more than 0.14 voltage is required to overcome the thermodynamic barrier. However, due to the internal losses, more than 0.2 voltage is needed. This small amount of voltage can lead to shorter digestion time by bioelectrochemical redox reaction and higher methane production than AD through direct or indirect methanogenic pathway. Ultrasonication is widely applied to wastewater treatment plants. Based on previous research, the high-strength ultrasonication is used for mechanical pretreatment to disrupt cell walls and solubilize it. However, low strength is applied for the anaerobic digestion itself. When applying low-strength ultrasonication during digestion, methane production enhances by increasing the mass transfer rate of organic matter, increasing enzymatic activity and cell permeability, so thereby stimulating microorganisms. So in this study, to enhance the MEC-AD performance, the intermittent low-strength ultrasonication was applied during digestion. With this research background, our hypothesis is application of intermittent low-strength ultrasonication to MEC-AD will increase the performance. Also, inputting the ultrasonic energy more frequently will achieve higher performance. In this study, we evaluated the effect of intermittent low-strength ultrasonication input at different intervals during MEC-AD batch operation. We operated these lab-scale MEC-AD reactors with an effective volume of 2 liters. This is a control reactor, and this is MEC-AD assisted with intermittent low-strength ultrasonicated reactor. The primary sewage sludge was used as substrate, and the inoculum was MEC-C sludge from previously operated in other reactors. The characteristics of sludge samples are shown in this table. In batch operation, the ultrasonic energy density was 0.005 watts per milliliter. And based on the on and off ultrasonic intervals, 1 second on and 29 seconds off, 59 seconds off, and 119 seconds off were labeled as US-MEC 30, MEC 60, and MEC 120 respectively. The normal MEC was controlled, and all MEC reactors were applied 0.3 voltage. This is a biogas production graph And compared to previous research with AD, MECs produced 90 to 105% more methane than conventional AD. In MECs, methane production was 5.6 to 8% higher in MEC with low-strength ultrasonication. In US-MECs, the methane production was enhanced as the interval longer, which was opposite to our hypothesis I mentioned before, by around 5,800 milliliter per liter. However, the methane production did not significantly increase when longer intervals were applied. This might be due to that the interspecies electron carriers such as hydrogen and formate are consumed by surface-reducing bacteria and methane simultaneously. The microbial community regarding exoelectrogen, SRB, and methanogen will be discussed in next part. Organic matter removal results were similar tendency to methane production, but showed a more noticeable difference. It implies that the intermittent ultrasonication stimulated the hydrolytic bacteria and increased the mass transfer rate, enhancing the organic matter degradation. In US-MECs, the highest organic removal was in the longest interval with 62% of VS and 62.2% of COD. The shorter interval could stimulate hydrolytic bacteria more frequently and increase mass transfer. However, it might be due to that after reaching the excessive level of ultrasonic energy to the microorganism, the performance enhancement is inhibited. I took the microbial samples from the bulk solution and I conducted the microbial analysis through ISIC 100 instrument. So in the microbial taxonomic analysis in bacteria, the major family level of bacteria was these 5. In the right figure of heat map plot, US-MEC 30 and 60 were grouped into one cluster. This implies a similar bacterial community population was observed at the shorter intervals. The positive correlation of Spirochaetaceae was observed in MEC, Sulfurfaceae was in US-MEC 30, and Syntrophaceae and Syntrophomonadaceae were in US-MEC 60. Spirochaetaceae is one of the syntrophic acetate oxidizing bacteria, SAOB, and Sulfurfaceae, Syntrophaceae, Syntrophomonadaceae include both exoelectrogens and SRB in genus levels. So the total exoelectrogen and SRB population changed from 21.6 to 28.1% when applying low-strength ultrasonication, and as the interval was shorter, the relative abundance increased. In archaea group, I separated these 3 major archaea, the diet partner methanothrix and hydrogenotrophic methanogens, Methanospirillum and Methanobacterium. This implies that the major methane producing pathway of MEC was indirect electromethanogenesis. And as the interval shorter from 1 seconds on per 119 seconds off to 29 seconds off, the methanothrix population increased from 35.2 to 42.5%, implying that the major pathway in US-MECs was changed from indirect to direct electromethanogenesis. Through this result, methane production of US-MECs was higher than MEC. In the literature, direct electromethanogenesis could enhance the methane production. However, in this study, contrary to the literature, methane production increased as the population involved in direct electromethanogenesis decreased. It might be due to the excessive levels of ultrasonic energy, and the SRB consumes electrons and electron carriers and reduces to hydrogen sulfide. The methane production was not significantly increased. The highest methane production was observed in US-MEC 120. This might be due to that both direct and indirect methanogenic pathways were enhanced and the less consumption of electrons and electron carriers was occurred in the longest interval. So conclusion, when applying intermittent low-strength ultrasonication, first, MEC-AD performance was enhanced. Second, the longest interval showed the best performance. Third, the relative abundance of exoelectrogen and SRB were enriched. And finally, as the interval shorter, the exoelectrogens and methanothrix population increased. It implies direct electromethanogenesis is the major methane-producing pathway in US-MECs. However, the longest ultrasonic interval showed the highest performance, so in future studies, it's necessary to elucidate the mechanism between intermittent low-strength ultrasonication and exoelectrogens and SRB to maximize methane production. Thank you for your attention. Any questions?